Students, now let's understand another type of operand system, and that is the repressible. Though it is missing from the NCERT, but it's a very easy one. Let's quickly go through it. Again by Jacob and Monod, this concept was explained. What the scientists found was, like for example, what is repressible? Let's understand that the operand is always functional. That means it is a part of anabolism because something has to be constructed. Catabolism is the destruction or breakdown. That is the lack operand. So in the repressible operand, it's a part of the anabolic pathway and generally continues to function. But what happens? It can be switched off if the end product is accumulated. That means the formation is in excess. The cell is not utilizing it that much. So if it is going in excess, it will be switched off. Otherwise, second reason it can be switched off is if it is made available from the outside. So what these scientists found regarding the tryptophan is an amino acid, and you know it's an essential amino acid. So in this operand there are 5 enzymes required. You can see here 5 structural genes have been mentioned. A, B, C, D, E are the enzymes prepared by them. So in the lac operand we learned about 3 structural genes and 3 enzymes. Here we have 5 structural genes and 5 enzymes. So in this operand 5 enzymes are necessary for the synthesis of amino acid called as essential amino acid. It's a tryptophan. These enzymes are synthesized by the activity of 5 different genes, and those genes are tri-A, tri-B, tri-C, tri-D and tri-E. Remember to go in the opposite direction. Now what these scientists found was if the tryptophan is added manually into the medium where E. coli is growing in the lab, so they found addition of tryptophan stops the production of the enzymes. Thus the system is known as the repressible system. Tryptophan when added, now what exactly happens is when tryptophan is added, it will attach as a co-repressor to the aporepressor and form a functional repressor. Now this is again according to 11th class concept of biomolecules chapter. There are some enzymes or there are some functional proteins which are non-able to function alone. That's why they are called as aporepressor. Now this aporepressor is synthesized by the repressor gene through messenger RNA formation, but it is not able to attach with the operator. So it is somehow different from the lac operand or inducible operand. Now this does not attach here, but it can attach here if it is made complementary with the help of the co-repressor. So if tryptophan synthesis is accumulated a lot, there is extra tryptophan or made available from outside, then this tryptophan itself will act like a co-repressor, attach with the aporepressor, form a functional repressor and then attach with the operator, and now this will be switched off. That's why it is called as repressible operand system. So you can understand there that for the switching off, either the product should be made available from the outside or the product is synthesized by the cell, E. coli in this case, and the product is tryptophan, and it has accumulated a lot, so it will give a feedback repression or negative control. Just imagine if there is too much of the food product, obviously it will send a negative signal that don't prepare more because there is already extra or excess is present. So that's why operand concept plays an important role. There are 2 more things which I have mentioned here: the leader sequence and the attenuator sequence. Actually leader sequence is present just next to the operator, and its function is to control the DNA segment which is called as the attenuator. An attenuator itself controls the rate of expression of these genes. So how fast should it happen? Because tryptophan is essential amino acid, should it be produced extra or less, or faster process or a slower process? All this is controlled by the leader sequence through attenuator sequence. I hope this all is concept is clear to you. Remember, all these operants will have negative control as well as the positive controls, and long time back I have told you negative control means when it will cause it to slow down, and positive means to activate more. Now next is, what is the advantages of having such operant system? So the main points I have already mentioned here are, first of all, to adjust according to the requirement. That means the cell is trying to perform homeostasis, or trying to save its resources, not to waste energy and time and resources for extra formation or less formation, or otherwise if there is no lactose, by to form those enzymes for digestion of lactose. And second, if there is already tryptophan, by to synthesize more tryptophan. So the cell is trying to adjust according to the requirements. Second point, it is economical, as the synthesis of the enzyme when required takes place. Otherwise enzymes are not synthesized, so it is economically better, or you can say the cell is trying to save its resources, time and energy. Next is this inducible or repressible operant system helps in growth and differentiation. That means what? If some genes are made inactivated, then obviously this will have a different effect on growth and development. And if some genes are activated, this will have again effect on growth and development. You can talk about many topics of the human body also. If a gene expression is controlled, like puberty, when we enter puberty there is change in the gene expression, and then suddenly you will find some boys, they start growing very tall, or otherwise some remain short itself. So genes expression, whether they function or not function, or they function more or they function less, this all is going to have growth and differentiation controlled. Next advantage is related genes can be switched on and off simultaneously. So I hope this all is clear to you. Just remember, induction is switching on, repression is switching off. Induction is done by inducer and repression is done by co-repressor. Induction is a part of catabolism, repression is a part of anabolism, and induction brings about transcription and translation, but repression stops the transcription and translation.